All right. Just let me know when to get started, Carol.
Um, Whenever you think is good, you can start.
Okay. Ready, Dr. Nath?
Yes.
All right. Hello, everybody. My name is Katie Lord. I'm one of the chief residents in the neurology program here, and I'm thrilled to be able to introduce Dr. Nath again today to give our grand rounds here at Mass General. So just a brief kind of introduction to him. He trained at the Christian Medical College in Ludhiana, India, and then completed a residency and fellowship in neuroimmunology and neurovirology at the University of Texas Health Science Center. He then completed a second fellowship in neuro-HIV at the NIH and has held faculty positions everywhere from University of Manitoba to University of Kentucky and Johns Hopkins. Currently, Dr. Nath works as the clinical director of the National Institute on Neurological Disorders and Stroke and is the chief of the section of infections of the nervous system. So he's been a pioneer in the fields of neuroimmunology and neuroinfectious disease, and his research has shed light on the neurologic implications of infections such as HIV, Ebola, Zika, and most recently COVID-19. He's published over 400 scientific papers on topics from the pathogenesis of HIV entry into the brain, to the role of endogenous retroviruses in ALS, to therapies for previously untreatable disorders such as progressive multifocal leukoencephalopathy. He has received numerous awards, including the Pioneer in Neurovirology Award and the NIH Director's Award in 2019. And this year has been featured in interviews with the New York Times, CNN, BBC, and Neurology Today. And I asked him for kind of a fun fact or the most underrated virus to share, and we discussed how there's not nearly enough research on rabies in this day and age. So now more than ever, I think we can truly appreciate the clinical and scientific impact of your work. So thank you so much for taking the time to be here, Dr. Nath, and welcome.
Thank you very much, Katie, for that very kind introduction. And so this is a repeat performance. Yesterday, I gave the same talk at the Brigham, and so I hope the audience is, except for Katie, so I apologize to you, you'll be listening to it twice. Okay, so I'm going to just launch into the talk itself. This building that I'm showing you here is building 10. So those of you who may not have been to NIH, this is the Clinical Research Center, and this is where all our patients come with we brought them with Ebola, with all kinds of, with COVID and all kinds of, every pandemic, we brought in patients over here. And it is a great place to do research because your labs and the patients are all in the same building, and it's very easy to go back and forth from one to the other. Okay, so I have no disclosures, and then I was asked to include this slide, and I'll just leave it on for a few seconds here so everybody can see all the conflicts. Okay. And I want to start with this quote by William Osler, who gave the keynote address to the American Medical Association in 1896. And he started off by saying that humanity has but three great enemies: fever, famine, and war. Of these, by far the greatest, by far the most terrible, is fever. And that holds true even today, because you know, the number of deaths that occur from each of these pandemics far outweighs anything where all the wars combined don't cause as much death and destruction as infections can cause. But however, we're very poorly prepared to handle any kind of pandemic. And this is at the time of the Ebola pandemic on the cover of Times, warning us that we are not prepared for the next pandemic. Okay. And Fauci has repeatedly brought to our attention that every president in the United States faces a pandemic. Despite all of that, the current pandemic we are in the midst of it, and we find ourselves totally ill prepared to handle it. And that's the saddest part of the entire story. And the other most important aspect that I want to drive home is that most pandemic infections have neurological manifestations, which are a major cause of long-term morbidity. So these pandemics will, the acute phase will go away, but then it's the neurologists that are left holding the bag as to how to deal with all these patients who have all kinds of neurological complications. But yet we don't have a plan in place, even as neurologists, how to handle these patients. And just if you look at the recent past, we've had a lot of pandemics. The Ebola, Zika virus, dengue, every year kills millions of people. You have acute flaccid paralysis, which is a polio-like illness. You have about a couple hundred children every other year. And yet, for any of these things, we have really nothing, and no plan in place. And this has consequences. And so some of us remember very vividly, and that with the AIDS pandemic, we didn't know how to handle it, and there was a lot of social unrest. So when you have large proportions of people affected by a disease, it can have huge implications. And that made a huge difference. The FDA changed the way they approved drugs, and new drugs were put together. A lot of effort was put together in trying to make a vaccine, although to this day we don't have a vaccine for HIV, and we don't have a cure for the virus either. But it has become a manageable disease. If you go further back in time, you'll see the influenza and the 1918 and 18 and 19 killed about 30 to 50 million people. That was huge amounts of deaths occurring in a span of just 2 years. But if you look at other viruses and just compare this to COVID right now, and you will see that COVID is, within the last 100 years, is the worst of pandemics that we've ever faced. Because even at the height of polio, at the peak times, there were about 40 to 60,000 deaths per year. Okay. Measles is an extremely deadly disease. At its peak in the United States, there were 3,000 deaths per year. With COVID, you have 3,000 a day. Just yesterday, 3,000 people died. Smallpox, if you go further back in time, you'll see that in the 1500s, the invaders, when they came to Mexico, 2/3 of the indigenous population died. Only 2 million were left. And when the European settlers came to Massachusetts, again, about 20 million people died from smallpox. Okay. And this is what smallpox looks like. This terrible disease, and invades all the internal organs, causing death. Now, if you go further back in time, we think that there were waves and waves of plague from the 500 AD time on, and then in the 1300s, there was another great plague of London that started in China. And it is said that these infections killed about 50 million people in the Middle East and Asia, 25 million people in Europe again. And but we don't really know for sure what the infectious organism was, right? And there was no microbiology in those days, so we are extrapolating that maybe this was plague, but we have no idea. There are probably some pandemic infections that kill people. But what is important is that these infections killed huge populations, and very few people were remaining. Then they repopulated the globe. For example, in England, you can trace the current Caucasian population to 25 families because everybody else got wiped out. And so human evolution in some way has been taking place through waves of pandemics over hundreds and thousands of years. Now, if you want to go back to hundreds of thousands of years, it becomes even more difficult to find out what types of infections prevailed at that time. One way to do that is to look at the human genome. And what you find in the human genome is that they are retroviral sequences. 8% of the human genome consists of retroviral sequences. 35% are these small bits and pieces of the virus that jump around the human genome, and they're called retrotransposons. And you can look at the endogenous retroviruses in the genome of non-human primates, and you'll find that the difference between humans and chimps is the acquisition of a endogenous retrovirus. Otherwise, the protein coding region is very similar. 99.9% is the same between humans and mice. So if you're 99% similar, what really separates us? What separates us is really the endogenous retroviruses. That's what really separates one species from another. And but yet, very little attention has been given to these viruses. Most of the effort is really spent on 3% of the human genome. When you say whole genome sequencing, it really is a huge hole in the genome, because what you're really studying is just 3%. Okay, that's a misnomer. So for the purpose of this talk, I'm going to address three major questions. The first is, what are the neurological consequences of pandemic viruses? How do these viruses invade, persist, and transmit within the brain? And lastly, what is the role of viruses in brain development and evolution? And here I'm going to talk about evolution of the brain itself. So the first one, again, I'm going to start with the most recent pandemic and go all the way down to the endogenous retroviruses. Here you have, almost 70 million people are infected, 1.5 million deaths. And within the United States, we're right now 3,000 deaths per day. I mean, this is huge. We haven't faced a pandemic like that in over 100 years. And yet, we've become numb to it. And that's the saddest part of it. And these numbers are underreported. So the reality is probably double of what we have here. Infections could be even 10 times as high globally. You never know. Yeah. And in the United States, it's really sad because of all the countries in the world, we have the most infections. Now, if you look at coronaviruses, there are 7 human coronaviruses, and each one of them causes neurological complications. Okay. And the closest to SARS-CoV-2 is the SARS-CoV-1, which uses the same receptor. So we've known for a while that these things can cause neurological complications, yet it took us quite some time to realize that SARS-CoV-2 actually has neurological complications. A lot of people were still debating whether it really does cause neurological manifestations or not. But what you can do is you can divide them into 2. One are the para-infectious manifestations that occur at the time of acute infection, and the post-viral ones that occur after the acute phase, and those are all immune-mediated. But it can affect the entire nerve axis. So the brain, spinal cord, and peripheral nerve can be involved. And it's an interesting phenomenon that still hasn't been fully resolved, is that some patients may be developing Ondine's curse. I think that's very fascinating, and we've certainly found a neuronal destruction in the respiratory nuclei of patients at autopsy. But so this is a study by Igor Korolnik from Chicago, and what they looked at was hospitalized patients in several Chicago hospitals put together, and they found that about a third of these patients have altered mental status, which is independent of the respiratory disease. Okay. And so it's not due to hypoxia. There are other factors in play here. And the encephalopathy is more common in older patients, and the neurological symptoms are more common in the young individuals. Okay? Because these, I think, are largely immune-mediated phenomena. So there's several types of encephalitis that have been reported. One is the viral encephalitis. This is the rarest form. In fact, you don't find much virus within the brain of these patients, but rarely you can. And so this is a patient who presents with generalized seizures, signs of meningitis, and you can find virus within the CSF. Now, there are many case reports of acute disseminated encephalomyelitis. That's not surprising. With most viral infections, you can get it. And here, this is usually a T-cell mediated phenomenon, or predominantly T-cell mediated phenomenon. And you'll see—
Dr. Nath, I'm so sorry to interrupt you. The slides are not advancing. Would you mind maybe stopping and resharing? Okay, they just advanced for me. Can someone in the chat say if that just worked? I know.
Okay, so let me do one thing. I probably am on my VPN. Let me see if I can get off my VPN.
I think it's working now. Should be good now.
I'm going to just quit my VPN. Like that's the culprit here. It'll take a little bit, and I'll come back again.
Okay.
And okay, yeah, here we are. Can you see the slide now?
Yes, we see the encephalitis slide, so I think it should work now.
Okay, good.
Thanks.
And so what I was saying is that acute disseminated encephalomyelitis has been reported with this, and there are many cases of them. But it's not surprising because most viral infections will cause that. But there are few cases of acute necrotizing hemorrhagic encephalopathy. And this is a fascinating immune-mediated phenomenon. We think it's largely mediated by cytokines, but they are bilaterally symmetrical lesions in the thalamus. That's how it usually starts. And then there's hemorrhagic transformation over there. You will get lesions elsewhere in the brain too, but there's usually some symmetry on both sides. And that's why we think that this is likely a cytokine-mediated phenomenon. Okay, so now this is neuropathology published by a group in Germany. They've published two papers, one in Lancet, another one in Nature Neuroscience. And what they point out is that you see a fair bit of inflammation in the brain, which is largely composed of CD68 and CD8 cells. Okay. They also claim that you find some virus, but the virus that they find is actually very, very low copy numbers. And I don't think that that alone can really explain the pathology that you see. We have a paper that should be coming out in New England Journal of Medicine very soon. That shows that there's a fair bit of microvascular pathology that's occurring in these patients as well. And you can, a lot of patients develop stroke as well. About 1 to 5% of hospitalized patients will develop stroke. And oftentimes there's more than one artery that is involved. So here in this patient, you can see bilateral strokes. And there's also some patients will develop cerebral hemorrhages. So you can see a lot of micro hemorrhages throughout the brain. What is also interesting about the strokes is that oftentimes you get venous and arterial occlusions at the same time. You can also get multiple organs involved at the same time. And here are some autopsy cases that we've looked at from the New York Medical Examiner's Office. And you can see, microhemorrhages and congested blood vessels in the brain. We also did MRI scanning of these autopsy tissues. You can see these punctate white matter lesions. Most of them are vascular lesions here. This is on a 7 Tesla magnet, and this is on an 11 Tesla magnet. You can see these blood vessels really well, and there's actually hemorrhage around it. That's why it's all dark over here around these blood vessels. So this is a group in Ann Arbor, Michigan, and what they did was they took a serum from patients with COVID and injected it into mice and showed that you can actually form a cargolopathy there. And these patients have antibodies directed against phospholipid antibodies, against phospholipids. So this hypercoagulable state is actually quite common in these patients. It's not entirely clear why that occurs. They do have a variety of other autoantibodies as well. And the current guidelines are to anticoagulate these patients prophylactically once you admit them for this reason. But as neurologists, our biggest problem is going to be, even if we have a successful vaccine and we vaccinate everybody, is that we're going to be faced with this long haul COVID. There are millions of people who continue to have recovered. Most of them are healthcare workers, and they have recovered from the infection. Oftentimes they even had mild infection, wasn't even serious illness. But 2 weeks later or so, they develop these myriad of symptoms. That includes persistent low-grade fever, probably due to vasodilatation, I think. Chest pain. It's an interesting chest pain. They get all kinds of MRI scans and of their chest, and can't find anything, but yet they complain of chest pain. My feeling is it's probably muscular in origin. They complain of sleep disturbances. The fatigue is incredible. The exercise intolerance is so profound that a cardiologist that I talked to, she said that just one flight of stairs up to her office upstairs, she could not handle it, and she had to lie down for hours to recover. And she quit her practice, was trying to do telemedicine, and even then couldn't get through the day seeing patients. So it really is a very strange phenomenon. It's been overlaps with chronic fatigue syndrome. And a lot of patients used to be called functional. I don't think they're functional, they have real disease. But we really don't understand the pathophysiology of this disease at all. It occurs 4 times more in women compared to men. So the key points with SARS is that the viral invasion of the brain is quite rare. There's a lot of microvascular injury that occurs with the infection. And that's because it invades the endothelial cells. And there's a lot of immune-mediated phenomena. And I think as neurologists, we need to recognize them because these things are treatable. And the earlier you treat them, the better off you are. Okay, so now I'm going to move on to Ebola. And so Ebola and Marburg have erupted in Europe and in Africa since the early 1900s. But we get few epidemics, few people get infected, and then it disappears. But in 2014, there was a huge pandemic, involved multiple countries, and it devastated West Africa. It is the virus is spread from bats to humans, and it's said that actually it's resident in pythons, and from pythons it goes to bats, and then from bats to humans. And as humans have been invading the territory of bats, we're contracting a lot of infections from bats. And it is amazing how we share so many pathogens with bats and not with other types of mammals. And the most likely reason is because humans lived in caves a lot longer than we've lived outside of caves. And so hundreds and thousands of years that we lived there, we've shared the microbiome with them, and we have a lot of genetic similarities. And these viruses have adapted to bats and humans in that sense. So at the peak of the pandemic there, you can see they had no infrastructure to handle these patients at all. And this is a picture by New York Times. And what they show is, people are dying here in this ward in various stages of death. And all they had was bleach here that they put on the floor. This powder is bleach in order to control. And this woman has all this vomitus and fecal material all over the place. And so people were fixated on the fact that this is a GI illness. And in reality, the neurological complications were not recognized early on. And so I went to Liberia at the peak of the pandemic because we started to realize that there were neurological complications and try to characterize them. And so we had an index case at NIH. This was a nurse from Sierra Leone. And when we admitted this patient, it was our ID folks realized very quickly, and Dr. Fauci was the attending physician on this patient. And he called me to say, Avi, can you come see this patient at 6 o'clock in the evening? Because he suspected that the patient had meningitis. And nobody had recognized neurological complications till then. So when I took a look at this patient, you had to wear all this garb to get in and get trained. It's a huge ordeal. But anyways, this patient had florid meningeal signs, okay? And he had generalized weakness. He was hemiparetic on one side, and his eyes were deviated to the right. So any neurologist would look at it and say, yes, this patient has CNS complications of the infection. And but we had no access to MRI. We couldn't take these patients into MRI scanners or even bring in pins or reflex hammers and the like in there. So you had to do the best you could. You couldn't even look in the fundus of these patients. But nonetheless, this patient received the best possible care. He actually recovered. And he had some minor neurological issues for quite some time, but now he's gainfully employed. Since then, we brought a number of patients to NIH from Liberia, and we've scanned them. A lot of them have punctate white matter lesions, okay, all throughout the brain. We've now established a cohort of about 200 patients in Liberia. And so at the peak of the epidemic, when I came back to NIH, I talked to my neuroimmunology fellows and said, was anybody interested in going there? And to my surprise, everybody said they wanted to go. And so all of us went down there, about eight of us, and we went down in turn. We established a cohort, and we're following them now. And John Bellew is the person who's really managing them currently. And so we followed them for a period of time, and you can see that they got better, but not all the way. So they still have a wide variety of neurological manifestations that persist. We also looked at a macaque model from Fort Detrick. So they do a lot of bioterrorism research there. And so this remains unpublished. But to our surprise, what we found was there's a lot of virus in the endothelial cells. That was not surprising. What was surprising to us was that we found restricted viral replication in the neurons. So a complete virus was not being formed here, but this VP40 antigen was expressed at very high levels in these neurons. So that came to me as a surprise, because that means that there's a reservoir in the neurons. of this virus. And so I'm going to discuss this kind of restricted viral replication, which is unique of what happens within the brain with a number of viruses in more detail. So the key points here are that neurological complications are frequent and are long-term sequelae, but are not recognized till later in the pandemic. Okay, but people get focused on the acute manifestations, which are often not neurological, and they miss all the neurology. And Ebola can form a reservoir in neurons with restricted viral replication. Okay, so next, moving on to Zika virus. And so this is spread by mosquitoes. The Aedes mosquitoes are found in these regions, and you can see that it spread very rapidly throughout the Americas. Although it's been endemic in parts of Africa and Asia for a long time, and it wasn't causing, it would cause some acute fever-like manifestations, but never really the kinds of neurological manifestations that occurred more recently. And that's because the virus mutated and evolved. And so the Zika virus was first identified in the 50s as causing a mild fever and rash. But in 2007, there was an outbreak in Yap. 100 cases, no neurological manifestations were reported. 2013 is when people started realizing that there were these huge increase in Guillain-Barré syndrome. So this is an interesting virus because the first indication that there was a new virus circulating was because of the development of Guillain-Barré syndrome. And so then they realized that there was a new virus, and this was the Zika virus. And then later, we started to realize that there were all these congenital malformations occurring in children. But that was much later because the gestational period is a total of 9 months. So it took much longer to realize that there were fetal complications. And that is because the virus infects neural progenitor cells, okay? And so much like rubella, it is able to cross the placental barrier and infect neural progenitor cells. It can also infect astrocytes and neurons. So patients will develop microcephaly or lissencephaly or acephaly. And so all kinds of congenital malformations can occur in the brain. However, some patients can also develop meningoencephalitis as well as myelitis. So you can get the whole spectrum of infection with Zika virus. And so there's a Zika forest in Uganda, and it's right on your way from Kampala to the airport. It's really halfway there. And so I was in Uganda trying to investigate nodding syndrome, which is another interesting pandemic, but I'm not going to talk about that right now. But on that trip, right by Lake Victoria, there are these beautiful labs that the Uganda government has, the MRC from England, and there's an NIH lab there too. They study HIV there. But I met this guy, Julius, and he's a virologist. And what he does is he has graduate students, and their job is to collect mosquitoes from all over Uganda. And they extract the blood from these mosquitoes and then sequence everything. And it's a very clever technique because when you sequence the entire genome there, you will find the organism that the mosquito is carrying. You will know what type of mosquito it is because it has the mosquito genome there. And you will find out what kind of animal the mosquito has been feeding in because you have those cells in there as well. So what he told me was that there were 243 circulating arboviruses at that time in Uganda, which is amazing because most of them are probably infecting animals and not humans, but they have the potential of jumping to humans. So I visited the Zika forest. Most of the forest is torn down now. There are just remnants of the forest left. So I had to have the driver position the camera so that it looks like I'm still by the forest, but most of it is just housing there. They've preserved, so you can see here, the other thing is that Zika, I realized they spell it locally with 2 eyes, although in the English literature now, one eye got dropped somehow. And but you will find this contraption in the forest that they've still preserved. And just look at this. This has these platforms here, and this was built by Alexander Haddo, who was a Scottish virologist in his times, and in the 40s and 50s, he built that thing. And what he did was that he put little cages here, and the cages he would put various types of animals in there. And then he would draw blood from them every day and then try to identify viruses. And what he found was that the mosquitoes, there were different types of mosquitoes you would find at different heights of the canopy. And so if you're a mosquito and you like to bite mice, you're at the bottom. If you feed on birds, you're on top. And if you feed on monkeys or something, you're in between, or humans, right? So all these had their favorite animals that they fed on, and they carried different types of viruses. So in 1948, he had actually identified Zika virus, right? And he had done a lot of very interesting virology on the virus, but identified it, cataloged it, and then forgot about it. So we got involved in Zika virus a few years ago at the time of the pandemic. And the approach that we took was, we said that we're going to try to see if we can develop a antiviral drug for this disease. And we decided to target the protease, because all viruses have their own protease. Okay? So just like HIV has a protease, and they're very unique to that virus. So they don't resemble human genome, so they're really good targets for drug development. So Rachel Abrams in my lab cloned out the protease genome, expressed it, developed an assay, and then in collaboration with NCATs, we screened a large number of compounds in many different ways. And so we had protease assays. We had, there were 2 different protease assays. We had a vero cell infection assay and neurodegenerative cell infection assay. And one of the things that we realized very quickly on that, the vero cell assays are not a good assay. And that's what traditionally everybody uses, because these have the PGP pumps that are very active in these cells. So you put in drugs and they'll get pumped out immediately. So people miss very important drugs in screening assays that way. So we dependent upon, we have human neuroprogenitor cells that we make from iPS cells, and we can grow them in large numbers. And so we use that as our primary screening assay. So that's why we discovered drugs that other people had missed. And one of the things we discovered was methicillin. And so that's a tetracycline that is FDA approved, but it's very potent in inhibiting Zika viral replication. We showed that in an animal model as well as in vitro. And so this paper just came out last month in PNAS. So we think that methicillin can be used prophylactically in patients who are going into high-risk regions, especially women of childbearing age group. So the key points here are that the Zika virus has acquired unique mutations to infect brain cells in utero and in adults. It can cause an immune-mediated syndromes, and methicillin has the potential of being a therapeutic as well as a prophylactic agent, both at the same time. Okay, so now moving back in time, I'm going to talk to you a little bit about HIV dementia, okay? And so in the pre-antiretroviral era, dementia was a major problem. And that's how my career got started. When I was a resident in Houston, my first rotation was through MD Anderson Hospital, which is the cancer hospital. And all AIDS patients were admitted over there. We didn't know that it was due to a virus. They had all these opportunistic infections. You had to glove and gown and go and see these patients. And as a resident, you had no other choice. My attendings never went into the room. And so, but I was very fascinated seeing these patients because, these were, highly educated men, and they were getting demented. And from the time they developed signs of dementia, they would die within 6 months, and they died a neurological death. And this is an MRI of a 30-year-old patient with HIV infection. You can see massive loss of tissue in the frontal lobes, as well as in the basal ganglia here, but the posterior parts are relatively spared. And on a PET scan here, you can see increased activity in the basal ganglia, because that's where you find a lot of inflammation and viruses in the basal ganglia. And then over a period of time, the brain just kind of shuts down. Right? There's no activity here in the brain. The entire brain gets involved. So the question is the virus in the brain? And if so, where is the virus? There is plenty of virus in the brain. And there are two cell types that get infected, and they have a totally different life cycle. So this is a fascinating virus because it infects CD4 cells. It's a cytopathic infection. CD4 cells die, and that's why the CD4 cell count goes down. Okay, but there are no CD4 cells in the brain. So what it is infecting are these macrophages. These macrophages are in the perivascular regions here. Okay, and they will form multinucleated giant cells. And this is an important reservoir because here it can produce productive viral infection. You can produce lots of virus out of these cells, but it doesn't kill these macrophages. Okay. So it's a very important reservoir for the virus. And the virus is adapted to infect these cells. It has to mutate to infect macrophages, okay? Because it has to adapt to CCR5 as a co-receptor. The other cell type that remains a little bit controversial, but in my way of understanding is there is no controversy, but people keep arguing all the time, is the infection of astrocytes. Okay? So in astrocytes, and it is really fascinating, but here is a restricted viral replication. So you don't get complete viral production over here. You get overproduction of the NEF protein, but you can find the viral genome there just fine. Okay? Shown here by in situ hybridization, the genome is there. Under certain circumstances, when you stimulate it with cytokines, you can produce a fully replicating viral particle, but under normal circumstances, it doesn't. But if you're a virus, this is a beautiful place to hide, because once you kill off the cells that you replicate in, you're going to not survive any longer. If you go into the brain, you can hide here for extended periods of time, until the lifetime of the individual. But in order to do so, the virus has to mutate. So if you look at the viral sequences in the brain and you isolate them from patients who have HIV dementia, the viral sequences are quite different, and especially for the envelope. Okay, and this is the consensus sequence right in the middle, and you can see that all these patients who have had ND means no dementia, had means HIV-associated dementia. They are the most divergent strains over here. So how does virus get into these astrocytes? That's also quite fascinating because it gets through by cell-to-cell contact. And cell-to-cell contact is very important for viral transmission because you can evade the immune system when you do that. And so here's a lymphocyte here in this yellow golden color, and in the blue is an astrocyte. Now the astrocyte has all these processes that it produces over here. And you can see it just produces these processes and they wrap around the lymphocyte, and at the point of junction between these two cells, these red dots are all these viral particles. So you have a lot of virus that is released right at the junction of these two viruses. This is by a scanning EM. And if you look at the same thing by transmission EM, what you find is these areas of contact right here, and there's virus that's budding off the lymphocyte and then touching the astrocyte cell membrane. What is critical is that the virus hasn't bud off the membrane, okay? So it's still attached to the lymphocyte, and it is touching this membrane at the same time. That contact between these two is absolutely critical for viral transmission to take place. If the virus buds off completely, it will not enter the astrocyte, okay? And so in this cartoon shown over here, as you can see these contacts, and this is just a composite of this over here, but this phenomenon is absolutely critical for viral transmission. I'll explain that to you why. And that is because the envelope is a trimer like this. It forms these petals, and when it's still attached to the lymphocyte, these petals are open. It's like a little flower, it's open, okay? Once it buds off the lymphocyte, it's called a mature HIV, these petals close, okay? And when it's in the open state, there is part of this GP120 or the envelope that interacts with CXCR4. You can only do it in the open state. A mature virus has to bind to CD4 in order to open this, and then it will bind to CXCR4. Okay? The astrocyte has no CD4, and that's why people keep arguing that the astrocytes cannot get infected. But you don't need the CD4. The reason you don't need the CD4 is that when you have the cell-to-cell contact, at this point in time, this is still open over here. And if it is open, it can bind to CXCR4. So the immature virus can bind to CXCR4. This fusion occurs. The fusion occurs, it'll just inject the material right inside the cell, and infection takes place. The other thing is that even with antiretroviral drugs, and we have this cohort we've established at NIH that have been on antiretroviral drugs with no detectable virus for over a decade. Okay, so they are the best control you possibly can have. No virus in the CSF, no virus that you can detect in the blood. Yet, when you look for TAT protein, you find a third of them have very high levels of TAT protein in the nanogram quantities. It's easily detectable by ELISA. Okay. And if you look at the brains of some of these patients, you will find that in these macrophages, all this brown stuff is TAT protein. And this can have all kinds of effects in the brain. Cause glial cell activation, it can cause neuronal injury. So the progressive changes we see in these patients, despite antiretroviral drugs, is driven by restricted viral replication. And this restricted viral replication is absolutely critical in the long-term consequences of HIV infection that are not controlled by our current antiretroviral drugs. Because once the virus is integrated, the only drug that acts are the protease inhibitors, and the protease inhibitors don't prevent the formation of all these other proteins, because they're formed very early on in the viral replication right here. So they get released even when protease inhibitors are there. Protease inhibitors only inhibit the GAG pole envelope precursor protein. They don't prevent any of these proteins from being formed. So the key points here are that the transmission occurs by cell-to-cell contact between lymphocytes and astrocytes. The virus mutates to adapt to the brain, and by doing so, it evades neutralizing antibodies. And there's restricted viral replication in long-lived cells in the brain that leads to persistent viral reservoirs. Okay, so now I'm going to spend a few minutes on measles. So this is not going back further in time. But for neurologists, I think this is absolutely important. You'll find measles on your boards at least quite often. Okay, so we had a recent outbreak of measles in the United States because we had people who didn't believe in vaccines any longer. And so there were about 1,000 cases of measles in 2019. But even in the peak, we would get close to 700,000 cases a year. The number of deaths were only 3,000 per year. Okay, but what is fascinating is that the kinds of neurological manifestations, you can get several different types of neurological manifestations with measles. You can get an acute measles encephalitis. But what is interesting is the syndrome that can occur years after the initial infection, which is a sub-sclerosing panencephalitis. And panencephalitis because it involves the gray and the white matter. And it causes progressive mental deterioration with myoclonus. And in the CSF, you'll find oligoclonal bands and intrathecal synthesis of antibody to measles, but there is no virus in the CSF. In the brain, is loaded with virus, you find nothing in the CSF. And I think that is very important because what I'll show to you, there's restricted viral replication, and that's why it never leaks into the CSF. And you'll see this classical EEG patterns on your board exams, these periodic spikes and sharp waves, 1 every 3 seconds. Okay, so here on the MRI, you can see gray and white matter involvement. And what I want to draw your attention to is this immunostaining for measles antigen and all these dots that you see here as this is in the axons here. And you can see it in some oligodendrocytes also over here. And shown in this cartoon, we think that the infection occurs in oligodendrocytes, causes demyelination, and it also gets transmitted by cell-to-cell contact to the neurons. And then it can transmit from neurons to neurons. And I'll show you how that can occur. So the way that happens is that the measles virus has to mutate in order to adapt itself to the human brain. And it acquires 2 types of mutations. The first one is in the fusion protein, which is the F protein here. And so all the sequences that were isolated from patients with SSPE, you can find these very unique mutations. And what it does is it increases the fusogenicity of the virus. The second type of mutation that occurs is in this M protein, which is the matrix protein. And mutations here prevent the complete virus from being formed. And you need both of these mutations for SSPE to occur. Okay. And so traditionally, when we think about transneuronal migration, we think of rabies and herpes viruses. These are fully replicating viral particles that go across the synapse. However, with measles, what happens is the fusion protein fuses the cell membranes together. So it's not going across the synapse, it's going across cell membranes, but after a fusion takes place. The matrix protein prevents the complete virus from being formed. So you get the RNA granules that are being formed that are traveling with RNA and proteins together from cell to cell. And that's why none of it leaks out into the CSF. So I'm going to show you the same example that this is not unique to measles. Here, this is with dengue, and we published this patient last year that we saw at NIH. And so this individual was from New York, originally from India, and he'd been traveling to India repeatedly to take care of his ailing father there. And but he developed a progressive neurodegenerative disease. Okay, and you can see this is a brain of a 30-year-old patient, and he had massive loss of brain tissue, both cortical and subcortical. He had oligoclonal bands, and people suspected viral infection. They did all kinds of PCRs, including all the arboviruses, never found anything. At the time of death, we found there were microglial nodules. So we suspected there must be some kind of virus. We took his CSF and did screened a phage display library. And what we found was that there were huge amounts of antibodies against 3 different types of dengue virus, serotype 1, 2, and 3, but nothing against serotype 4. When we looked at the brain, we found immunostaining for dengue virus, there was virus everywhere. The brain was just loaded with virus. And we found it in endothelial cells, we found it in neurons, we also found it in astrocytes. And here by in situ hybridization, we can show you here in these neurons have the RNA for the virus. And when we sequenced it, it was all dengue serotype 1. So what we think what happened was that this patient was infected with 3 different serotypes, but only 1 was present in the brain. The virus formed a persistent but restricted viral replication that was efficiently transmitted throughout the brain, but did not leak into the CSF. And it is the repeated viral infections that I think was key in this patient. So for viral entry to take place, the antibodies will then bind to the virus and drag them into the cell. And that is a very scary thing because this can happen with other viruses also. And we don't know if that's the case with coronavirus. We certainly hope that's not the case. Yeah. Now, the last one in the last 5 minutes I'm going to talk about is these retroviruses. And these endogenous retroviruses have been absolutely critical in brain development. And as you can see, as we go across the species, one thing that changes is the size of the brain. And the only other thing that changes is the acquisition of endogenous retroviruses. So they are absolutely critical in brain development. And the way we acquire them is that all retroviruses are sexually transmitted. And the only reason they're sexually transmitted is they're trying to get into the germline. Once they get into the germline, then the new offspring that is formed is the equivalent of a transgenic individual. And because once the virus gets in there, gets into the genome, then it cuts and pastes itself throughout the human genome. And so it acquires all kinds of new functions that it never had before. And you can find these things, and then even during the process, even during brain development, when you go from a neural progenitor cell all the way to neurons, there are somatic mutations that take place. So these line 1 elements, which are transposons, they will keep jumping through the human genome. And so the line 1 elements of an oligodendrocyte are going to be very different than those of a neuron, and very different at the stage of neuronal stem cells, okay, because they are jumping somatically at each stage of differentiation. And this is a beautiful paper by Chris Walsh, who showed that even in the adult brain, if you look at different places within the brain, you will find different line 1 elements scattered there, showing that and the properties that we develop in different areas of the brain is driven by these retroviral elements. And this is the most recent virus we acquired was this endogenous retrovirus K, and it sits right next to the proline dehydrogenase and is maximally expressed in the hippocampus. So they're differentially regulated. And so a lot of hippocampal function is probably driven by an endogenous retrovirus. And this is a nearly complete retrovirus. We found that it was expressed in stem cells here, and in very early stages of embryonic development. And as these stem cells differentiate into neuronal stem cells and then to neurons, the HERV-K gets shut down. Okay. But it's very critical in maintaining the stemness of these cells. And unless it gets silenced, brain development will not take place. Okay. We published a paper in PNS earlier this year where we mapped the entire pathway as to how these endogenous retroviruses maintain the stemness. And there's a very unique secondary signaling pathway that is critical in this process. And there's the receptor that it uses is CD98. So there's very scant literature in the role of these endogenous retroviruses in brain disease. So as you can see that if some of them are absolutely critical in early embryonic development and they're shut down for a particular reason. If you reactivate them, they can do bad things. If you reactivate them in cells that can undergo mitosis, like breast tissue or prostate tissue, you'll develop cancer. If you push neurons to mitosis, because all retroviruses are oncogenes, neurons cannot undergo mitosis. They'll just degenerate. So it's no surprise that neurodegenerative diseases could in part be driven by these retroviral elements. And we've just started scratching the surface of those possibilities. But if you look at these diseases, there are different endogenous retroviruses that people have implicated in each of these diseases. And I'm going to talk to you a little bit more about ALS because that's what we've been studying in our lab. We looked at the genome database from the New York Genome Center and we downloaded the entire RNA sequences that they had available on these patients from autopsy tissues. And we categorized them into 4 different groups. Controls that had no activation of these HERV-K loci. And then we had about 200 some individuals here who had multiple loci that were activated. And then there were other groups that had either none or very few. Okay. So if you look at this, what we call as ALSA, you can see that compared to controls, they had activation of this endogenous retrovirus, what we call HERV-K, and chromosome 22 and chromosome 19. So there are 2 different loci in chromosome 19. And they were highly activated in this patient population. And the other interesting thing is that in collaboration with Merit and with James Berry, they were very kind to provide us all the samples from the NEALS consortium. What we found was that when you measure this in serum of patients, the HERV-K levels, the duration of disease is what is critical. So the longer the duration, the higher the viral load. But it's not age dependent. So it's not just because they're older that you get more virus. It is dependent upon duration of disease. We made a transgenic mouse model with it. And what we found was that if you just take the envelope and express it under a neuronal promoter, you're a classical ALS, upper motor neuron and lower motor neuron involvement. And it's better than the SOD and TDP43 transgenic mouse models because you get both motor neurons involved. The survival rate is about 50% at 10 months, so they can be easily studied. And they develop all the pathological features, including aggregation of TDP43 in the cytoplasm of these mice. So the question is then, how can you explain that ALS gets this anatomical transmission that occurs, progression that occurs in patients with ALS going from one part of the body to the other? And I think the answer is here in this beautiful study looking at this ARC1 protein, which is absolutely important for synaptogenesis. And what they found was that this is actually a gag protein, okay? And it binds RNA and will go across the synapse. And so we have started wondering about the same possibility that maybe the gag protein of HERV-K can just take the HERV-K virus and viral genome and transmit it. And I showed you with all these other viruses that there's restricted viral replication with measles, with dengue, all these things occur the same way. And we suspect that that might be the exact same phenomenon that's probably occurring here, that the so-called RNA granules are the mode of transmission from between neurons to neurons here as well. A group in Australia did treated patients with antiretroviral drugs. We screened antiretroviral drugs against HERV-K, and they're not that great, but they do have some effect. So they found that over 24 weeks, these patients with ALS, and we did all the viral loads here in a blinded manner, the viral load does come down in serum, but never comes down to zero. But what is fascinating is you take these patients off the antiretroviral drugs, and the viral load rebounds within 1 week. It's back to where it was. So that, I think, is definite evidence that these drugs are acting directly on HERV-K. And I think this is the first in vivo demonstration that that is really the case. I'm going to not go over this data here in the interest of time, but what I want to say is that retroviruses have been critical in human brain evolution and development. Reactivation of these viruses or somatic insertions in terminally differentiated cells, such as neurons, can cause neurodegeneration. I think that's really important. That's the unique aspect of the brain, that you can, if you activate any oncogene in the brain, you cause neurodegeneration. They likely form RNA granules and get transmitted across neurons. They can be targeted by antiretroviral drugs, but we really need to develop much better agents if we really want to control these viruses. So in summary, neurologists need to be prepared to handle acute and long-term consequences of pandemic infections. We need to study virus-brain interactions, and that can provide us unique insights into neurodegenerative disease. And academia needs to play a major role in developing treatments for persistent violence. We cannot depend upon pharmaceutical industry for this purpose. And these are the people who actually do all the work that I've shown you from my lab. And this is my lab as well as the clinical team that's been doing all this work. And then all our collaborators, and particularly want to highlight Merit and James, because they've been very instrumental in supporting our program and helping us throughout it. I'll stop here and take any questions that you might have.
Thank you so much. And I did just put in the chat Zoom information for 1 p.m. kind of open forum research discussion organized by Dr. Oaklander. It also should have been emailed out to all MGH faculty and fellows. So thank you for agreeing to do that as well. There is one question in the chat. I'm sure more may chime in. Dr. Yenny Lanoya asked, is there a reason why viruses tend to hide in the stem cells of the brain? I believe this has also been seen for herpes viruses like CMV and HSV1 and Zika. What is special about the stem cells?
Yeah, so the stem cells express a lot of genes. They, the more primitive the cell, the more chromatin is open and a lot of things are available. So viruses take advantage of those things because now they're easy targets. The only difference is for the virus to get to them. If you can get to a stem cell, that's where it wants to go. That's the best place for it to hide, because stem cell will differentiate to some place and it'll carry the viral genome with it. So it's not surprising that viruses will attack stem cells. And retroviruses, they like to get into germ cells. They can get transmitted that way too.
And feel free to let us feel free to let us know. I know it's 10:01, but there are a couple more questions if you have just 1 minute. Dr. Staley asks, says, fascinating talk. Is it possible that HERV-K activation is a non-specific reaction to neuronal stress akin to herpes reactivation?
Yes. So we've looked at that possibility. And so at least I can tell you what we've done. It doesn't answer the question completely, but provides you some insight. So if you take neurons and you put mitochondrial toxins on them, glutamate on them, hydrogen peroxide on them, HDAC inhibitors on them, you don't see activation at all. You have to, we've been able to activate it using CRISPR-Cas9, and we've been able to activate it if you take demethylation and HDAC inhibitors. You have to do them both together. And it makes sense, because if it was easy to activate like that, if it's by stressing, the human race would be extinct. You won't exist any longer, because these things cause havoc. They cause cancer, they will destroy neurons, the whole bit. So they are very tightly regulated. And so they're not that easy to activate.
Dr. Brad Hyman also asks, I'm curious what other RNAs might hitchhike with ARC across the synapse. Any thoughts? Is this normal physiology?
Yes, I think there's normal physiology. So the ARC protein is so critical for synaptogenesis. That's normal physiology. And there are probably lots of RNA granules. We just have started to realize that this is an important phenomenon. People have started talking about RNA granules in the context of neurological diseases, but I think it's an important physiological function. The viruses only hijack normal physiological function. They are just so small, they have very little over the genome. That's the only way they can survive. They don't have an ability to create new pathways and new mechanisms. They just co-opt the existing mechanisms. Yeah.
And maybe one last question. Dr. Kim Kegel asks, has HER-VK or retroviral elements been implicated in rare developmental disorders?
That's a very good thought. I don't think anybody has looked, but I wouldn't be surprised. The problem is that sometimes, they've caused such massive problems that so with all the spontaneous abortions that take place, it's quite possible a lot of these things go wrong over there, so that the fetus just never matures or survives. But one could collect tissue from spontaneous abortions. I think you could probably learn a lot about these viruses.
Okay, if anyone else is interested in ongoing discussion, there's the meeting at 1, but I think we'll conclude our grand rounds for today. Thank you so much for being here. I can say I genuinely enjoyed your talk twice, so that says something.
All right. Next time you can give it.
Yeah, do one, see one, teach one, right?
All right. Thank you so much.